Thanks for joining the TMAU webinar here in June 2015. My name is Rob Platika, and I'm a community manager on rareconnect.org with the European Organization for Rare Diseases, eurordis. Today's webinar is brought to you by the TMAU community on Rare Connect, and we've done a few different webinars with researchers who are interested in TMAU. So this webinar continues in that series. And we're excited to have Jake coming to talk with us today. If you haven't joined yet, please take a moment to sign up and join in the global conversation on managing the symptoms of TMAU on the TMAU community on Rare Connect. So Jake Wintermute is a postdoctoral researcher at Paris Descartes University, who today is going to be presenting synthetic biology and microbiome engineering approaches for Trimethylaminuria, TMAU. Jake is a biologist by training at the Center of Interdisciplinary Research in Paris. In the summer of 2014, Jake challenged a team of undergraduate students to use genetic engineering to improve human health and well-being. The result was The Smell of Us, a collection of new biotechnologies to control body odors by engineering the bacteria that live on human skin. And you can find more about that online, and Jake will go through that today as well. Also in this webinar, Jake will discuss his group's work in progress on TMAU. The primary pathology of TMAU is the appearance of trimethylamine in the sweat. Many microbes are capable of metabolically neutralizing TMA, including some that occur naturally on human skin. Genetic modification may enhance this ability, creating microbial products to alleviate the symptoms of TMAU. Jake will discuss his scientific progress and the specific challenges of developing a genetically modified organism for human use. If you have questions while Jake's presenting, please type them in the chat box, and we'll try to collect them and save them for the end of the presentation, where we'll have time to try to get some of your questions. Okay, thanks very much. I'm now going to throw it over to Jake and pull up his slides.
Great. Thanks, Rob. It's really, I'm happy to be here. Yeah, if anybody has any questions during the talk, go ahead and type them into the chat box. I'll do my best to answer them in real time. Although I'm a novice with this platform, so it's possible that I won't know what I'm doing. So hello everyone. My name's Jake. I'm a biologist. I'm a postdoctoral researcher. I'm new to this particular area. I'm new to trimethylaminuria, and I'm new to the community. It's been great meeting some of you, and I hope to meet more of you in the future. I think you guys have a really great community, really effective community, and it's something that's very I think it's just great to see. So today I'm going to be talking a little bit about my research and about my students' research. The work I'm describing is kind of a scientific work in progress. So I don't know how common it is on eurordis to share scientific research at this relatively early stage. It's something that I think it's a good idea. It's something that my institute values in particular, is trying to bring in the public, trying to bring in patients' associations into research at an early stage. But it's something that is not too common in the popular media. So it's much more common for scientists to present their research after it's published, after it's tested. And so that's not the case for the work that I'm going to be discussing here. So it's important to kind of know that and take that with a grain of salt. The work that I'm describing, it's a work in progress. It's not a proven scientific fact. Okay, so with that said, let me get started by introducing myself and my institute, and how we got interested in body odor, and specifically in TMAU. So this is us. So we're the Center for Interdisciplinary Research in Paris. We're an interdisciplinary research group. If you're familiar with American universities, we are we're quite a bit different from an American university. So we're a place in Paris. That includes researchers from many different disciplines. We have a lot of interest in education. We have a lot of interest in public outreach, um, and we're loosely affiliated with Paris Descartes University, but we're a relatively uh, independent institution. So the CRE unites researchers from a variety of different fields. Uh, we have a kind of three-part mission. So we do basic research at the CRE, and that's a lot of what I'm going to be talking about is basic research. Um, so we're a biology lab, kind of like you would find a biology lab in the United States or at another major research university. We do teaching, so we have undergraduate, master's level, and PhD level uh, degree programs that grant degrees through Paris Descartes University. So we're a teaching institute, and I teach at the CRE a course called Introduction to Synthetic Biology for master students. And we also have uh, a mission of uh, public engagement. So we're interested in what is sometimes called citizen science, where members of the public can become actively involved in scientific research, either in generating data or in analyzing results, um, or in proposing research questions. So we think that there's a lot of value and also a lot of scientific power in getting the public involved in research, and so that's something that we try to do whenever is possible. Okay, so that's our group. There's in total there's maybe 100 people who are sort of loosely associated with our group in one way or another, including about 20 active biology researchers. So now let me talk a little bit about the team that was behind the research that I was going to present. So most of the actual lab work was done by master students. So our team included master students, included PhD students, postdocs like me, and included professors like my boss Ariel Lindner, who worked on this project either kind of independently, just because they came to us and thought it would be an interesting thing to work on, or because they're students in our master's program and we're doing this work for course credit, or for reasons of their own. So a lot of the people who worked on this project were not even biology students. So we worked with industrial designers and graphic designers and web designers and product designers and biologists and chemists and so forth. All worked together on this project, and the most of the results I'm going to present were produced last year over a period of about 6 months, as a project being presented in what's called the International Genetically Engineered Machines competition, or the iGEM competition. So the iGEM competition is I've got a little the website here is a good reference if you want to see more detail about the work that we did, or the team, or just the context for the project. So the iGEM competition is an undergraduate research competition. So there's more than 300 teams of undergraduates from all over the world come together. They spend their summer working on these genetic engineering projects or biological engineering projects, and they present them at MIT at the end of the summer, and it's a lot of fun, and there's prizes, and there's a lot of enthusiasm because they're undergraduates and so forth, and sometimes very good work comes out of this. Very innovative work comes from these young people. So This particular work won 2 prizes. It won the Best New Application and the Best Art and Design prize. And I think if you go to our website, you'll see in particular some of the sort of the excellent designs that the students came up with to communicate the work. Okay, now let's get to the work itself. So, in this project, we're focusing on the skin, right? So the inspiration for this project is the skin, and in particular, it's the skin microbiome, right? So your skin is not your skin, right? Your skin is full of life. It's like a rainforest, right? On every person all over the world has a skin that is full of living, metabolizing bacteria. There are millions of species, millions of different strains of bacteria that are, contributing to almost any function of the skin that you can imagine. Bacteria are playing a role. This is something that scientists have known for a long time, but not really been able to study effectively, in part because a lot of these bacteria can't be cultured in the lab, but they can be studied with newer techniques involving DNA sequencing. So they can be studied through DNA sequencing techniques. And so it's only very recently that scientists are coming to appreciate the kind of diversity of microbes that live on the skin and all of the roles that they play in the sort of health and normal functioning of the skin, right? And so I'll give just a little taste of that on the next slide. So this is from one of the first really comprehensive studies of the skin microbiome, right? And so this is just a purely descriptive study. They're going in, they're sequencing a bunch of DNA, and they're asking just what kinds of bacteria are there, right? Just like you would go into a rainforest and count the animals in the rainforest, right? So a very preliminary, right? Just the first thing that you would do, right? What bacteria are there? And they find millions of bacteria, millions of different strains. They come from all of the different branches of the bacterial tree of life. There's proteobacteria, there's cyanobacteria, there's corynebacteria. So it's extremely rich, and it's extremely varied, extremely diverse, complicated ecosystem that's going on in your skin all the time, right? So to me, like, it's hard to sort of conceptualize what's going on just because of all of the complexity and all of the diversity. So to me, I think of the microbiome by analogy as a kind of living thing, something that will respond to outside perturbations, kind of like a living thing would be, kind of like its own organism. I think of it sometimes like a rainforest, just this rich, lush environment full of thousands of different species that are all interacting in complex ways. The truth is that nobody really knows all of the different roles that are being played by the bacteria that live on our skin, but there's just so much activity that we know it has to be important. And so I think in the next few years, we're going to learn a lot more about the bacteria that live in our skin and everywhere else on our bodies, and all the different roles that they play in our health. Okay, so that was our motivation. There's millions of bacteria on the skin. What are they doing, and how can we use them to improve human health? So the topic that we decided to focus on was body odor. So it turns out that human sweat by itself is naturally odorless. So all of the molecules that we perceive as natural body odor are produced by the metabolic activity of bacteria on our sweat. So the bacteria, they consume the sweat and they produce a range of different molecules that we perceive as body odor, right? And so one sort of obvious consequence of this is if you have no bacteria, that you have no body odor. And so this is, for example, how conventional deodorants work. So conventional deodorants, they contain antibiotic, antibacterial ingredients that kill the bacteria that naturally live on the skin. Because you don't have the activity of those bacteria, you don't have the smell molecules being produced. So that's a very straightforward application. So normal deodorants, they work in a very general way. It's you know, if you think of your skin as a rainforest, a deodorant is, you know, is paving over the rainforest, right? It's killing everything that's there, and leaving no living thing behind. And as a result, it eliminates these conventional body odor molecules. But so that's the kind of simple level of understanding, right? But the more we looked into natural body odor, the more complexity we saw, right? So body odor is produced not just by one molecule, but by hundreds of molecules, and not just by one strain, but by hundreds of strains. The perfumes, the odorant molecules of body odor, they can give a different impression depending on their level or how they're combined, right? So, for example, the same molecule that might give an unpleasant odor, like a goat smell or a rancid smell, at high concentration can give a pleasant odor at lower concentration, a musky odor or a fruity odor. And this is also true depending on how the different components are combined and the overall odorant profile. And so here the best metaphor that I can think of is to a wine, right? To a fine wine, right? It's not there's not just one molecule that's contributing to the odor of a wine, and the you know, the French people love it when I make this metaphor, right? But there's a whole range of possible odors that are detectable, that combine to give an overall effect, and it might be a pleasant effect or it might be an unpleasant effect, um, depending on the subjective presence of the person perceiving it. And so this is the body odor wheel that I'm showing. This was made by a designer who at this website, and this is a kind of a direct analogy to similar odor wheels that exist for wine. Okay, this is the situation that we face as scientists, right? So first of all, we have all of the complexity of human sweat. Then we have all of the complexity of the microbes that are acting on the sweat to produce the odorant molecules. And then we have the fact that body odor itself is effectively subjective in the way that it's perceived, right? It's subjective. It simply is, right? So a robot, right? A machine, a gas chromatograph can't tell you if a body odor is pleasant or not. And if you want to improve body odor, right? If you want to understand body odor, there's no simple chemical formula that can tell you, is this a good body odor? Is this a bad body odor? Have we improved body odor? You know, with this new deodorant product that we've invented, right? So the fact is that only human smellers, only human beings can tell you if a body odor is pleasant or not. There's just no other way. And so that brings us to the kind of the citizen science aspects of this project. So we designed an experimental platform to better understand how the composition of bacteria on our skin contributes to the body odor in our sweat. So excuse me. So the platform looks like this. We go out to the public, right? To citizen scientists. We collect their sweat samples, including the microbes inside of them. We take them back to our central lab, cut them up into pieces, right? Cut up the cotton pads into pieces, give them to human smellers who can give us their subjective impressions over whether this is a good body odor or a bad body odor or not. We plate out the bacteria in the sweat samples on petri dishes to collect a sample of the bacteria that are present in the sweat, and in that way we start to, first of all, we build a kind of library of data on natural human sweat smells. And we also build a library of strains, a collection of bacteria that are present in sweat that may affect body odor directly or indirectly. Okay? So this is the platform that we came up with. So it was kind of it's nice for us in the sense that sort of human smellers they're much cheaper than expensive gas chromatography equipment. But it turns out that the human nose is extremely sensitive. It can detect odor molecules at parts per million concentration, and it really is comparable to many of the finest instruments in terms of its ability to detect smells, right? And only a human being can tell you if a smell is pleasant or not. So we did this over the summer. We went out to the public. We collected their sweat samples in glass tubes like this. So people wore cotton pads under their arm to collect their sweat. They exercised for a little while. They told us about their lifestyle. They told us whether they were wearing deodorant, what was their diet like. We collected information about their age and their gender. And then we divided the body odor samples and we gave them to other people to smell, right? And so we asked them to score the smells on two major axes: the intensity of the odor and the pleasantness of the odor. So the scoring was done on an online platform that looks like this, right? So this is the smellofus.org, and it's a website, and you can go there and you log in and you can you put in the code for the smell that you're smelling, and you can say you put in information about your lifestyle and your diet. You say, is this a weak odor? Is it a strong odor? Is it pleasant or unpleasant, and so forth? And we collect all of the data, and we build for each odor this kind of graphical presentation of an individual subjective impression of the odor, right? So how did it make you feel? What was the emotional response? What did it remind you of? Did it remind you of a fruit smell or of an animal smell? Did you like it or not, right? And so all of this contributes to the kind of citizen science aspect of the project, because so first of all, we want to reward the users who are donating their sweat samples by giving them a good impression, or giving them an interesting feedback about their personal smell. And we want to make the process fun for the people who are essentially working for us for free, the citizen scientists who are taking the time out of their day to smell and rate other people's body odor. So some of the results that we got from this are quite interesting. So, for example, we we didn't notice any difference in the body odor profiles of men and women on average, which was quite interesting. We noticed that people who use deodorant have a slightly worse body odor than people who don't use deodorant on average, which was quite surprising. We noticed an overall positive correlation between the intensity and the pleasantness of people's body odor. So the stronger someone rated a body odor, the more likely they were to like it. So people preferred strong body odor in this context. And we also collected just a wide range of very interesting subjective impressions. So this graph is showing a plot of intensity versus pleasantness on a scale of 1 to 100. So the points in the upper right-hand corner are the strongest and the most pleasant body odors that we detected. And then the words are just free association words that the smellers spontaneously came up with to describe what they were smelling. And so one sample in particular was just seemed to get consistently high ratings for both intensity and pleasantness, and it was described as the Colombian beauty. And so this is one of the samples that we decided to focus on in greater detail to try to understand, what were the microbes that were contributing to this Colombian beauty odor. Cool. And so I think so we collected a lot of data and so forth. I think the main lesson that we took from this exercise was that I think this is something that is really interesting to the public. I think this is something that we can really hope to get the public involved in an active way, right? So everyone is curious about their own body odor. They can't smell themselves. They don't know how other people perceive their body odor. And they want to know. They're curious about it. So we collected overall more than 300 samples, and people were just very excited to see how they smell. So I think that this is a project that we're trying to scale up. This is something that we can really reward citizen scientists for participating in, because we can give them sort of interesting, detailed feedback about their specific body odor that otherwise they have no way of knowing, right? So we can say, how does your body odor compare to an average person? Is it more intense or less intense? Is it more pleasant or less pleasant? What does it remind people of? Does it is it a fine wine? Does it remind people of cherry or gasoline or butter or cheese, right? What is this thing that you're sending into the world? People are very curious about it, and I think that I think it's the kind of thing that can really draw them into the process. So we can so here on the left, these are our little social media icons that we generate to describe each individual smell, right? So this is like you can share this on Facebook, right, or something, to compare your body odor with your friends, right, and in that way bring other people into the process. So that's what we can give to the public. I think we can give users lots of interesting and valuable data, but we also get a lot out of it as scientists, right? And in particular, what we get out of it is the strains, right? So we have a huge library of strains now, right? Bacteria collected from hundreds of different people with different lifestyles, different ethnicities, different ages, different genders, um, from among just all of the millions of strains that existed on their skin. And these are, in general, they're unknown to science. They're completely unknown to science, right? So these are strains that, um, no one knows. No, they haven't been studied. They're right. They're completely unstudied. People gave them to us freely, and they represent for us, as scientists, this just totally untapped resource, for understanding the function of the skin microbiome. And I think they also represent, to us as engineers and bioengineers, potential tools for improving the microbiome, or for kind of engineering the microbiome, to produce a desired effect. So somewhere in this library of strains, there are the good ones, right? Somewhere there are the strains that are producing the body odor of the Colombian beauty, um, or I think potentially that might have many other effects on body odor. And so for us, this is a sort of, it's a very valuable resource, and it's something that we can, convince the public, to give to us for free. Um, and so for that, I'm just gonna I think uh, I'll pause.
Okay.
Yes. Um, yeah, I so the short answer is I don't know. Short answer is I don't know. It um this does seem to be, the case, or roughly the case. So the more deodorant people use, their body odor is often perceived as being, um, less pleasant. Um, and the truth is that I don't know the answer. I wish I knew the answer in detail. But if I had to guess, I would use this metaphor as of a rainforest, right? So when you go in and you clear cut a rainforest, right? By putting antibiotics on your skin, for example, uh, and then, uh, let the rainforest grow back, right? It's not always the most beautiful species that come back first, right? So when you disturb, the ecosystem, right? It can be the weeds that grow back first. And I think a similar thing could certainly be happening in your skin, right? Anything, when you sort of upset the balance of the microbiome, if you wanna think of it that way, you can end up, favoring the growth of, um, strains that you don't want. Yeah.
I see another participant mentioned in our community, the TMAU community. It seems that we can't smell ourselves, even though it is metabolic. It seems to be the rule in any type of body odor or halitosis, and that is a topic that I think I've seen come up quite a lot. That people can't smell themselves, or sometimes even their close family members can't notice the smell. Is that
Yeah,
anything you've
yeah. I think this was very interesting to me. I was very surprised when I learned it too. And, yeah, I'd like to know more about it. So I'd like to know, for example, what do you do? Like, what strategies that do you use to evaluate your own, um, body odor? This is something that would be very, very interesting to me.
Okay, and maybe we can come back to that or other questions at the end
Yeah.
as well. Hopefully, we'll have a couple
Okay.
minutes.
Okay. So
Okay,
now,
thanks.
so let me press on, a little bit, and describe, uh, how we might be able to relate the microbes that live on your skin to TMAU as a condition. Okay, so, here's TMAU. I'm sure you're all, more or less, familiar with it, so TMA originates in the stomach as part of the digestion of choline, trimethylamine. Normally, it's delivered to the liver, where it's metabolized by an enzyme called FMO, flavin monooxygenase, into a compound called trimethylamine N oxide, which is odorless, right? So TMA is responsible for the fish odor, but TMNO is odorless, and normally this metabolism happens in the liver. In TMAU, the FMO enzyme is not available because of a genetic mutation, and so as a result, the TMA becomes systemic, and it's primarily secreted in the sweat and the other bodily fluids, and that's what results in the odor. So I think in the long term, prospects for the disease are probably in gene therapy, right? It's probably in using gene therapy to restore the function of this enzyme. I have no special insight into how long it will take for gene therapy to become clinically relevant, but it does seem clear that that's what you wish you could do, right? But in the absence of that, maybe we could restore the function of this enzyme by adding it to the natural complement of enzymes that are found in your microbiome, right? So the FMO enzyme, it's not at all unique to humans. It's found in many bacteria in nature, and so here what I'm showing is so this is work from our lab. So we took the FMO enzyme from another bacteria called Rugeria that expresses it naturally, and we added it into E. coli with genetic engineering, right? And so with kind of the modern state of genetic engineering is that it's very easy to take a gene from one bacteria and put it into another gene. So the bacteria on the left are expressing the FMO enzyme, and the bacteria on the right are not. And so you can immediately see that there's a difference in the color of the two enzymes. So the FMO enzyme being expressed in the bacteria on the left is so it turns out it's a very generally functioning enzyme. It doesn't just oxidize TMA; it oxidizes a range of other molecules, and in fact it oxidizes tryptophan, right, which is a naturally occurring amino acid, to produce a pigment molecule called indigo. So it turns tryptophan into indigo, and so therefore it's very easy, for us in the lab to see to detect the enzyme, because we can add tryptophan and produce this blue pigment. So the idea in a nutshell in this project is why not restore the FMO enzyme to a skin bacteria so that we can metabolize the TMA in the sweat before it becomes volatile and contributes to the body odor. So it's very useful for us in the lab to be able to follow the enzyme by watching this blue pigment. It is a kind of a funny coincidental side effect of this enzyme. So here these test tubes at the top, the ones on the right have the FMO enzyme, and the one on the left don't, and you can see the very rich blue pigment. And so you might ask yourself, well, if I put this enzyme you know, if I put it on my skin, if I put these bacteria on my skin, would they turn my skin blue? uh, and and the answer is, of course, we hope not. So indigo is it's a harmless pigment. Tryptophan is uh, not present in the sweat at a high concentration, and so uh, we don't think that this is a likely side effect uh, in using these bacteria in an actual treatment. Um, but there it is. Yeah. Is there a question? I thought I saw someone typing a question. I don't. Right. So, yes. Yes. Well, we hope not. We hope not. And we don't think so, right? But the truth is that we wouldn't know for sure until we tried uh, human trials. So the other possibility, so the beside so the so one possibility is we produce these GMOs, right? So we take a bacteria that lives naturally on the skin, we express this FMO enzyme, and then we add this to the skin as a kind of cosmetic product. So that's one possibility. The other possibility is to use the library of strains that we've collected so far and that we might continue to collect to do kind of bioprospecting for natural skin bacteria that express this the enzyme naturally, that express the enzyme naturally. So just in kind of browsing or searching through the genomes, so mining the genomic data of bacterial strains that are already known uh, to live on the skin, quite a few of them uh, express this enzyme naturally, right? And so the nice thing about this is that these wouldn't be GMOs. These would just be taking naturally occurring skin bacteria and adding more of them, right? Finding the ones that do this the best, um, and adding more of them. Um, the So that's the potential upside. The potential downside is that we may not be able to find a strain that expresses this activity as much as we could with genetic modification, right? With genetic modification, we can really crank the enzyme up to a very high level, higher than it's probably is found in nature. And so a GMO approach could potentially be more effective. So those are the kind of trade-offs that we make with that consideration. Um, so we've thought a little bit about how this might work in the real world. So this is our kind of our vision. So here's your armpit, your axillary vault. So here are the bacteria that live naturally on your armpit, including a naturally occurring strain that we want to replace. So, oops, yes. So we'll go in with our probiotic product. So we'll package our bacteria into a kind of a cream, uh, add them to your armpit or to anywhere on your skin, right, where they will replace the naturally occurring bacteria, right? And so they'll carry this FMO activity, and so therefore they'll be able to digest the TMA as it comes from the sweat. So I have this is an example of what the product might look like. So this is an emulsion that I prepared with some of our bacteria. So I'll see if I can show an example here. So here, this is what it looks like. This is just for an example, but I mean, I'm not a cosmetic chemist, but I prepared this with uh, bacteria, soy milk, beeswax, and jojoba oil. And so you can kind of see. I mean, it's a cold cream. It's a cold cream recipe. Um, it's an emulsion, right? It's not. I mean, it's like a cold cream. It's not slimy. It's like a pleasant product. It has no particular smell. It smells a little bit of beeswax. Um, and it's an emulsion, which means that it's mostly fat, mostly jojoba oil with drops of water, drops of liquid, um, sort of micro drops suspended inside. And so you can see. So here's a microscope image of what the cream looks like on a microscopic level. So the sort of the gray blobs are the fat layer, the oil. The black blob is the water layer, and the purple dots are bacteria, right? So the bacteria is living in these kind of micro dots, inside the emulsion, right, which allows them to be delivered to the skin, um, through like a normal cold cream product, for example. So that's our vision. That's our overall vision. I hope it's kind of clear, um, at least what we're thinking about. And I want to just conclude a little bit by talking about where are the roadblocks. So what's um, uh, oh, so there's a question here. I thought soy wasn't good for us. So I actually didn't know that. To tell you the truth, I didn't know it. Um, but it wouldn't have to be soy in this case. I was using the soy milk just as a neutral media to grow the bacteria. It could be almost anything. It's also the in this case, this because the soy milk's not being processed in the stomach, right? It probably wouldn't produce choline. Um, yeah, it's a good question. I didn't know that about soy. Um, okay, so I want to talk a little bit about the potential roadblocks, for kind of this approach, or this kind of approach, right? Um, so for us, so the first one, right, we have to be quite clear, is that we don't know that it works, right? It's not proven science, and there's really no way to know that it works, except trying it in humans, right? Um, there'll be no there'll be no proof. There'll be no proof that it works until we try it in humans. I mean, there's some little things that we can do to try to kind of simulate, but basically it needs human trials, to see if it works. So this is the major stumbling block, right? And we should be clear, like it's not we won't call it we can't, it's not the cure, right? We can't call it the cure. We haven't done the experiment. It's very early, much earlier than you might normally see, science presented in the popular press. So anyway, okay, I won't belabor the point anymore. But we don't know that it works. We hope that it works. So the other important stumbling blocks is, it's a GMO, and GMOs are complicated. They make it a lot more complicated to test. So this is why, basically, this is why I can't send it to you, right? I mean, it's not. I'm not a lawyer, and I don't know the exact regulations, but I guess that I would probably go to jail if I sent samples from the lab for human trials, right? It needs to be approved by an IRB. There needs to be oversight. We need to make sure we're complying with all the different regulations. There's going to be a lot of paperwork. There's going to be informed dissent, or informed consent, rather. Um, so all this stuff, right? And this gets to the next roadblock for us, which is that in our lab we don't do human trials, right? So we don't have experience with that. Like in a research hospital, they'll have they have a wing, right, where patients can come in and they can do their human trials, and they have the lawyers to manage the paperwork, and they have physicians who can run the trial. But we just don't have those things. We're not physicians, and we don't have the facilities to do human trials. And so this is a stumbling block. We need to find a partner for that. Some kind of a research hospital, or possibly a drug company, or possibly a cosmetics company, who can help us just manage the human trials. Uh, and and then the final thing is that it's all expensive, right? It's something like $1 million to do a basic human trial for a product, and we don't have $1 million in funding for this project right now. So we need to get that funding. We need to get it either by applying for grants, or by partnering with some kind of a drug company who can fund the relatively expensive process of making it actually happen. So that's it. That's it. So yeah, I don't want to, I don't want to end on a too negative of a note, but that's that's kind of how we are in science. We always have to be very careful with our caveats. But even with all those caveats described, I'm optimistic. I think it has a chance. It's something I'm willing to invest my personal time in. And it's something that I'm quite hopeful about, and I hope it's something that we can keep moving forward in the next few months. Okay, and with that, I will conclude, and I will say thank you for listening, and I will answer any questions.
Thanks very much, Jake. That was really interesting and a nice presentation of your work so far. So I see some people have a couple questions already,
Yeah.
including could we eat forbidden food, so food high in choline, if a treatment like this were successful?
Yeah. So that's a good question, and the answer is just that I don't know. They're the only there it's the way that you would answer that question, right? It's quite clear. You would take a group of 100 people. You would give them a bacteria, or you would give them a placebo treatment. You would tell them to eat choline-rich foods or not, and then you would rate their body odor after that treatment, and it would give you the answer. And that's when you do that, then you know the answer, and before that you don't. So that's where we need to get. Yeah.
Okay.
Yeah.
The next question is about research there and how people can help and what kind of partners you might be thinking about.
Yeah. So we don't have a partner yet, but we are looking. We have some nice connections through Meebo Research, for example, who are helping us to get in contact with other researchers in the community. So we're completely open about collaboration. We're not we're not worried about getting scooped, or we're not worried that someone is going to steal our idea, or that they're going to patent it or whatever, and then they're going to take all of our patent money. We're not worried about any of those things. We just want to do whatever it is to keep it going forward. Yeah, but we don't have a collaborator right now. So can you envisage it as an internal product? So you would love it. An internal product would be better, right? Like I think that's clear, why it would be better to do an internal product, because you can kind of get the TMA at the source before it's distributed to the rest of the system. And in this particular approach, we can't do an internal product with this approach, and the reason is because the enzyme requires oxygen. So normally this process happens in your liver, where there's lots of oxygen is coming in all the time in the blood, and so the normal version of this enzyme has lots of oxygen to work. But in your stomach, there's no oxygen. There's no oxygen in your stomach, and so if you, I mean, you could, right? You could put this enzyme in a pill and swallow it, but it almost certainly wouldn't be effective just because there's no oxygen. So we're looking for other. It would be nice to find versions of this enzyme that don't require oxygen. We're looking for them. It seems like it's going to be a little bit more complicated to try to do an internal approach. So it's not something that we're working on right now. Yes, body odor versus breath odor. So good question. So breath odor is another thing, and the kind of the short answer is we don't know. So your mouth and your back of the throat and the origin of sort of varies in terms of the presence of oxygen and the availability of oxygen, and it also varies in terms of the bacteria that live there, right? So at least in principle, you could do a similar thing with a bacteria of the mouth. You could add this enzyme to it, and use that as a mouth treatment. So some of the reasons why, some of the kind of unknown things about that are, is there going to be enough oxygen in the mouth to allow this enzyme to function? And we don't know. And then the other thing is, are there special regulations that apply once you're in the mouth, right? So like the kind of the more internal you get with your treatment, the more safety regulations, the more the harder it is, right, to conduct the human trial, the more cautious everyone has to be. And so like a skin treatment versus a mouth treatment versus an internal treatment, they might be more or less difficult to actually get approved. What do we have here? So what do I? So yeah, so this is a good question. So the question is what treatment would I recommend? And the truth is that I kind of have to turn that question around, and I would like to know what treatment you recommend, right? Because I think that for me, sort of coming at it as a scientist, right? Like I know how enzymes work and so forth, but the truth is, right? Like I in my talk, like there's very little that we really understand about the kind of the skin in practice, right? Like this is a I know textbooks, right? I know about textbooks, but this is a real world problem. And so one of the reasons why I want to reach out to the community is because I want to understand what you're doing, right? What strategies you're using, and I've heard some quite interesting ones so far. I mean, you know way better than me, right? Absolutely, right? And what I'm hoping is that I can kind of I don't, maybe just by understanding the kind of real world experience, I can help to sort of shape the scientific work that we want to do. I think it's definitely going to be the case for this, that it's you guys, it's the real world people that need to guide the research, and not the other way around, because the truth is that we the scientists, we know very little.
Oh, that was a good answer. Thanks. Someone was wondering if crowdfunding would be something you would consider, and would be something that could help the project along.
Yeah,
at any stage. I know, you mentioned one of the barriers was the high cost of drug development, and yeah,
yeah,
that's a huge one, especially if you're not a company, with the experience and the money behind to put behind trials.
yeah. I think so, the truth is I don't know. I mean, I think that it could be I think it could potentially be interesting. I don't know. I mean, the first thing you got to say, right, is that there's some ethical pitfalls in this kind of crowdfunding, and I don't know how you could avoid them. Like I mean, like, what if it doesn't work, for example, right? Like then, uh, did I just rip off a bunch of people, getting them to crowdfund my, you know what I mean? Like it doesn't, it seems ethically risky, to have the patient community sort of crowdfunding their own treatments, like just because of, I don't, it just seems, I don't know. Um, so crowdfunding per se, I don't know. Um, but I do like, I think so. One of the values of crowdfunding is that it can demonstrate that a market exists that people otherwise didn't know about before, right? And and that is something that this community can do, right? So not even necessarily with money, but just with voices, right? By saying, when we like, I mean, I think it's one of the reasons this community exists, right? Is you can is to kind of make your voice heard to the drug companies and say, we're here, we're a market, we're an organized market, we want you guys to invest money, in this area, and we're honestly, we're prepared to buy what you make, right? Like it's, I mean, that's what drug companies, for example, respond to, right? Like and I do think it's valuable, um, to show that a market exists in that way. Yeah.
Yeah, I do know of another clinical trial for another metabolic rare disease called alkaptonuria. Did a successful crowdfunding campaign, but it wasn't to get a trial started. It was once the trial was already started and approved, to help the travel costs for some participants who had to go from different countries around Europe to specific travel sites. And that's where I think you get a bit away from the ethical
Mm.
problems and questions that you mentioned, where maybe you can find other ways to use crowdfunding to support different pieces of the
That's
trial, but not the
yeah,
creation of it itself.
right. Yeah. Oh, I like that. Yeah, I like that.
Yeah. Ronnie had a comment here. I'm not sure if it's one that you can comment on. And, Ronnie, I know a lot of other people in the community have a similar frustration. Ronnie's saying that they're taking activated charcoal and copper, supplements recommended by the doctor, hopefully, and trying to change the diet to a low choline diet, but it's not working for them.
Yeah.
And wondering what the purpose is. And yeah, I mean, this is something that others have said too, and many try all the suggestions that come their way, and say still, despite trying dozens of different suggestions for months or weeks at a time, they haven't really noticed a difference. Is there anything you can kind of say to
Yeah.
those folks who are in that position?
Yeah. I mean, so all I can say about that is that I think that those stories are, I think that they're very powerful, and I think that they can sort of be an effective, uh, tool in the hands of the community, in terms of, sort of getting more resources, um, allocated to your problem. I think that, uh, yeah, I mean, I think that those kind of stories are. I mean, I think it's the essence of the community, and I think it's the it's going to be the most effective sort of advertising, or the most effective tool for sort of winning more attention and more resources to this community. Like I think if we succeed in getting our funding, if we are able to get a grant, or if we're able to partner with a drug company, it will be um, partially because uh, people understand, right, that it's like there you try all these things, and it like nothing is working, and the disease is totally neglected, and no researchers, or you know what I mean, like nobody, it's ridiculous. Honestly, like it's absurd. Like I've never seen uh anything like it. Just how I'm surprised as a biologist to see uh like frankly how neglected this disease is. It's I've never heard of anything like it. That there, uh, I mean, there's a handful of people, of scientists working on it, and it should be way more, and I think it could be way more, um, if we can, with the right attention.
Yeah, I just want to mention that if you're ever interested in doing any polling or surveying of the community, that we can help with, and work with Amiibo research on as well. That'd be something we'd be interested in, and I'm sure everybody attending this webinar would be interested in filling out a survey, or helping you to gather more information about things that they've tried that haven't worked, or things like that. And that's something we've done for a few other communities, and could create here.
Great.
Um, yeah. So someone's asking what's happening next about research. And I also wanted to mention too, before I forget, can people still take part in the Can You Smell Us website and project? I mean,
Yeah,
yeah.
that's discontinued for now, but we do hope to get that
Okay.
rolling again soon. Um,
Okay.
so the next steps for us are finding a partner, and finding funding. So this is the next thing. So I'll be going to conferences and presenting our work to try to um, get some more attention, and we'll be looking for grants targeted maybe to neglected diseases, and we'll be contacting potential industrial partners, maybe cosmetics companies, maybe drug companies, um, to see if we can, convince them that there's a profit opportunity here. Um, Yes. So I see the question is, there's a lot of people that are affected that aren't coming forward. So I think that this is true. Like uh I think that so right, it's a yeah, I think it's true. So this is a um it's a for example, it's a recessive disease, and the most severe forms occur when you have two copies of the mutation. But if you just have one copy of the mutation, you might have a reduced form of the disease, or a transient form of the disease. And I do think that there are a lot of people out there, and maybe they don't even know that they have this disease. And I think that this is something very this is a very powerful case, right? So for example, right, if I could convince a drug company that there's a million people, and I'm just making that number up, right? But a large number of people who have this disease and don't know it yet, or have a sort of transient form of this disease. Well, that suddenly that makes it much more attractive for them, right? If we could potentially attract much more resources, if we can convince them that there's this large invisible market. Yeah, and this trying to understand, it's another reason why we want to reach out to the community to try to understand, um, where these invisible people might be.
Okay. the next question is, and maybe people can kind of ask their last question, because I know Jake's been really kind to spend this evening with us so far, and probably has to get on with his night here. But someone was wondering if there's a website, or if you'd ever think about creating a website for the project. So it seems like something, an idea for the future.
Yeah, so we do. I mean, we have a the students made a website, and it's quite good. Um, I don't think that the current website doesn't it doesn't really communicate that the work is ongoing. And so that is something that we would like to do. Um, we don't have any immediate plans for doing that. I get so I guess I don't want to lose the opportunity to connect the community. I didn't plan any particular way. I believe that I am going to be joining a TMAU Facebook group, which is another way to get in contact with me. My email? I'm happy to get emails from anyone who wants
Okay.
to email me.
Yeah, I can share your email. You're also welcome to join the community on Rare Connect if you ever see anything that you'd like to comment on. Of course, we already have a page about this webinar, which we'll add to the video afterwards. So if you want to, pop in there and say hello there, people can get in touch with you. That's really great that you can make yourself available like that, and I'm sure some folks might have some more follow-up questions, maybe even after watching this webinar. So I'm just, is not that copyright? Just
That's correct.
put,
Yes.
yeah, okay, great. Yeah,
Yeah. So email
there's
me,
Jake's email.
and I'll join these groups. So I'm in the short term. I'm interested in hearing sort of personal testimonials, like what do you do, what works for you, and sort of why do you think it works. Those are very valuable. And then in the long term, we'll be we'll be um, we'll be sort of asking the community to sign a kind of letter of support as something that we can include in a grant application, for example. Yeah. So those are
Yeah,
the those
that's great.
those are the plans for now, and I'll definitely we'll we'll keep you posted.
Okay. Yeah, thanks very much for your time, Jake. This was really interesting, and I know everybody here in the room really appreciates it, and is really happy that there are some people interested in understanding and treating trimethylaminuria like you are. So, thanks. We'll post the video of this webinar at that link that I shared, which is the same link that had the information about the webinar. And yeah, if anybody has questions for Jake, they can reach him there on email, or make a comment on the webinar video, and we can pass it along to him. And yeah, we look forward to being in touch, Jake. Thank you very much.
Thank you. Take care.
Okay, thanks everybody. Have a really good night.